Have you ever stopped to wonder if the air you breathe, the water you drink, or even the place you live is somehow, well, shaping your health? There's an entire field of science that digs into exactly that, and it's called environmental epidemiology. So today, we're going to pull back the curtain and see how these incredible health detectives connect the dots between our world and our well-being.
So this is really the fundamental question, isn't it? Is our environment making us sick? And more importantly, when we have that nagging suspicion that it is, how do scientists actually go about finding the answer? Proving that link is the entire reason this field exists.
All right, here's our game plan. We're going to start with why we even need this field in the first place. Then we'll define what it actually is. After that, we'll get into a super important distinction, exposure versus dose. Then we'll check out the tools these scientists use, see some real-world examples, and finally we'll talk about the biggest challenge they face, which is the idea of certainty.
Okay, first up, a world of unknowns. You know, so many chronic diseases don't have a single obvious cause. And there's this growing concern, both from the public and from scientists, that factors in our environment are playing a much bigger role than we thought.
So, an investigation can really kick off from two different directions. On one hand, you've got a disease looking for a cause. This is what happens when a bunch of people in one area get sick with the same thing, and everyone starts asking, what is going on here? What's in our environment that could be doing this? But then you've got the flip side, a cause looking for a disease. This is when we already know a pollutant is out there, maybe from a nearby factory, and scientists have to figure out, okay, what health problems, if any, is this thing actually causing?
So who are these people piecing together the clues? Let's jump into our next section and really define their field. It's time to meet the health detectives.
First, let's zoom out a bit to the parent field, epidemiology. Now, at its heart, this is a science of patterns. It's not focused on why one single person got sick. Instead, it looks at huge populations to find trends, the who, what, where, and when of a disease on a massive scale. Think of it as looking at the health of the entire forest, not just one tree.
And environmental epidemiology, well, it narrows that focus way down. It's looking specifically at things outside our bodies that affect our health. Pollutants in the air, chemicals in the water, contaminants in the soil. And because these are things we often can't control with our personal choices, they pose a really unique challenge to public health.
All right, now we're getting to what is probably the single most important concept in this entire field. Seriously, if you get this difference, you get the whole shebang. Let's talk about exposure versus dose.
Exposure is pretty simple, really. It's just the opportunity for something to get into your body. You know, you walk into a smoky room. You're exposed to smoke. A chemical is in your tap water. You're exposed when you drink it. It just means the substance is there and there's a way for you to come into contact with it. But dose is the game changer. That's the amount of that substance that actually gets past your body's defenses and gets absorbed. Just because you're exposed to something doesn't mean you get a big dose. Dose is what matters. It's the amount that actually has a chance to cause a biological effect.
So how do scientists even try to measure exposure? It's not just a yes or no thing. They use this really elegant little formula. They multiply the intensity of the substance, so how much of it is there, by the frequency, how often are you in contact with it, and the duration, or how long that contact lasts. A tiny amount for a really long time can be just as significant as a huge amount for a few minutes.
Okay, so if these scientists are basically detectives, what's in their toolkit? How do they actually go out and gather the clues they need to crack these incredibly complex cases?
Well, since they can't and shouldn't do experiments on people, their main tools are these really clever observational studies. Take a cohort study, for example. They might follow thousands of people for decades, tracking their exposures over time to see who eventually gets sick and who doesn't. Each one of these study types is just a different strategy for finding those hidden connections.
And today, one of the most powerful tools in their arsenal is GIS, which stands for Geographical Information Systems. Think of it like a superpower. It allows scientists to layer maps of health data on top of maps of environmental data. It turns rows and rows of boring spreadsheet numbers into a visual story, letting them see things like a cluster of illnesses right around a specific factory. It's a total game changer.
But even with an amazing tool like GIS, you run into these tricky trade-offs. See, if you study a really large area, like a whole state, your population numbers are pretty stable, but you lose all the fine-grained detail about what each person was exposed to. But if you zoom way in on a tiny neighborhood, you get great detail on exposure, but your results could get totally skewed if just a few people move away. It is a constant balancing act.
Okay, enough of the theory. Let's open up a few real-world case files and see how all these methods have been used to uncover some absolutely critical links between our environment and our health.
First up, malaria and El Niño in Colombia. Researchers looked at decades of climate and health data and found this incredibly clear connection. During El Niño years, when it's warmer and wetter, malaria cases shot up by more than 17%, just like clockwork. This was huge because it meant they could use weather forecasts to predict malaria outbreaks and get resources in place ahead of time.
Next, let's look at temperature. A big study across 11 cities in the US found this perfect U-shaped relationship between temperature and mortality. It turns out the risk of dying goes up not only on super hot days, but also on super cold days. The safest, lowest risk spot was right in the middle, in that mild temperature zone. It's basically statistical proof that our bodies have a Goldilocks temperature range.
And finally, let's bring it inside our own homes. A massive study in Western Europe looked at the link between indoor air and asthma, and they found something subtle but powerful. Homes with higher indoor humidity consistently had more kids with asthma symptoms. It's a pretty stark reminder that our most personal environments can have a huge effect on our long-term health.
Now, these success stories are incredible, right? But we have to be honest here. This line of work is unbelievably difficult. So let's talk about the biggest obstacle they all face, the challenge of certainty. And here it is. The single biggest headache in this field has a fancy name, exposure misclassification. But think about it. It's actually pretty simple. How could you possibly know with 100% accuracy everything a person has been exposed to every single day for the last 30 years? You can't. It's impossible. Scientists have to estimate, and every little error in that estimate, saying someone had low exposure when it was actually high, can muddy the waters and make it that much harder to find a real connection.
And that really leaves us with this one big, forward-looking question. Our environment is changing faster than ever before. So how will these health detectives adapt? How will they refine their tools and their methods to keep up and to uncover the new connections that are going to be absolutely essential to protecting our health for generations to come?